So, have you ever wondered how scientists can pinpoint a criminal from like a single hair? Or how they detect a virus from a swab that looks totally clean? Well, the answer to both is this incredible technology called PCR. Today, we are going to unpack this amazing tool. It works on a molecular level, but believe me, its impact on our lives is absolutely massive.
All right, let's just dive right in. We're going to start with a problem. A problem that actually connects detectives, doctors, and even paleontologists. Think about it. Forensic science, virology, ancient history. These fields couldn't seem more different, right? But here's the thing. They all face the exact same fundamental challenge. And as it turns out, they all rely on the very same piece of technology to get their answers.
The problem, every single time, comes down to scale. I mean, whether it's a tiny drop of blood, a swab for a virus, or just some dust from an ancient mammoth bone, the amount of genetic material you start with is, well, it's practically nothing. You can't see it, you can't weigh it, so how on earth are you supposed to analyze it?
Well, this is where the genius comes in. There's a brilliant solution to this problem, an invention so profound that it just completely changed the game for scientists everywhere.
It's called the polymerase chain reaction, or what everybody just calls PCR. And really, all it is a way to amplify or make a ton of copies of one specific piece of DNA. This elegant idea came from a guy named Kary Mullis, and it was so revolutionary that it won the Nobel Prize in Chemistry back in 1993. Okay, let's forget all the technical jargon for a second. The absolute simplest and honestly the best way to think about PCR is like this. It is a super specific photocopier for DNA. Imagine you have a giant library full of genetic code. PCR is the tool that lets you find the exact page you're looking for and then print billions of copies of just that one page. And yeah, when I say billions, I'm not exaggerating. After just about 35 to 40 rounds of copying, a single starting molecule of DNA can be turned into over 10 billion copies. That's the magic right there. PCR takes something that was totally invisible and makes it so abundant that it's easy to see and analyze. So, how does this molecular photocopier actually do its thing? Well, it's not magic, it's chemistry, and it's a surprisingly simple and repeatable recipe that's all controlled by just changing the temperature up and down. So to get this reaction going, you have to mix a few key ingredients in a tiny little tube. First, you need your DNA template. That's the stuff you actually want to copy. Then you add primers, which are kind of like little molecular bookmarks that tell the machinery where to start and stop copying. You also need a special builder enzyme called Taq polymerase. And of course, you need the raw building blocks, the A's, T's, C's, and G's to actually build the new DNA strands. Now, this is where it all happens in what you can think of as a 3-temperature dance. First up, denaturation. You crank the heat way up to about 95 degrees Celsius, and that heat literally melts the DNA double helix apart into 2 single strands. Then you cool things down for annealing. This lower temperature lets those little primer bookmarks find and latch on to their specific targets. And finally, extension. You raise the temperature back up to about 72 degrees, which is the perfect temperature for that Taq polymerase enzyme to get to work, building brand new DNA strands. And here is the real key to PCR's incredible power. That 3-step cycle doesn't just happen once. Nope, it repeats 20, 30, sometimes 40 times. And every single time it runs, the amount of your target DNA doubles. So 1 copy becomes 2, 2 become 4, 4 become 8, and you can see how this gets out of hand really, really fast. It's pure exponential growth. Now, obviously, doing that precise temperature dance over and over again by hand would be, well, impossible. So, of course, scientists built a specialized machine to automate the entire process for them. And this is it, the thermal cycler. Its job is actually beautifully simple. It's a machine that's programmed to very rapidly and very precisely heat and cool those little reaction tubes, running through the denaturation, annealing, and extension steps again and again and again, perfectly, every time. You know, these machines aren't all one size fits all. A big, high-throughput model, for instance, might be able to process almost 100 samples at once, perfect for a huge research project. But then you might have a smaller, personal model that handles fewer samples but can actually heat up even faster. It just goes to show you how the hardware itself is engineered for all kinds of different scientific needs, from massive screening operations down to a single person's experiment. It's really important to get this.
PCR isn't just one single tool. Over the years, scientists have tweaked and modified the basic recipe to create an entire toolbox of related techniques. And each one is designed to answer a very different kind of question.
So one of the most important variations is called RT-PCR. You see, standard PCR starts with DNA. But what if you need to study an RNA virus, like the flu, or you want to see which genes are actually switched on in a cell? Well, then you have to start with RNA. RT-PCR is super clever because it works backwards. It uses a special enzyme to first turn the RNA into DNA, and then it starts the copying process. This one trick makes it an absolutely essential tool in modern virology. And the toolbox just keeps getting bigger. You've got quantitative PCR or qPCR, which doesn't just tell you if a DNA sequence is there, it tells you how much of it you started with. And then there's multiplex PCR, which cleverly looks for several different DNA targets all in the same tube at the same time. It's just a great example of how flexible this core idea really is. Okay, let's move on to our final section and take a look at where all this is headed. Because this powerful technology is getting smaller, faster, and way more accessible than ever before. The next huge leap forward is taking this entire process and just shrinking it. The goal now is to create a complete lab on a chip, integrating everything you need into a single tiny microscale device. So why does that even matter? Well, the goals here are pretty ambitious. We're talking about making the technology small enough for what's called point-of-care testing in a clinic, a doctor's office, maybe even in your own home one day. Making it smaller also means you get results way faster, you use tiny amounts of expensive chemicals, and you can make the whole system tough enough to be used outside of a perfect laboratory setting. And that really brings us to the future. I mean, from the mammoth bone to the doctor's office, PCR has already completely changed our world. But as this technology keeps shrinking, as it gets faster and more accessible to everyone, what new problems are we going to solve? What new questions will we even be able to think of to ask? That, right there, is the exciting future that this molecular photocopier is still writing.